This last section of the Patterns of Evolution chapter is on origin of life on Earth. Science probably never will know exactly how this happened, but we never will know exactly how photosynthesis happens either. Theories, though, are being developed based on research and observations about how life might have started on this planet. For the last 50 years, most scientists have been talking about the tidal pool method of um, uh, originating life, which is due to a reducing atmosphere. Chemicals in the atmosphere formed that came down in the rain to form puddles full of interesting biochemicals. When the puddles dried, they formed polymers, and the polymers then, when re-wetted, formed what are called coacervates or aggregates, which are clusters like soap bubbles of chemicals. The possibility is then that those gave rise to the first cells. The problem is, though, recent evidence indicates that probably Earth's atmosphere was mostly nitrogen and carbon dioxide, and they don't really do much in the way of monomer formation. So I'll talk more about that in a minute, but that doesn't look likely. Panspermia is the concept that we could have come from outer space. Living things could have come from an asteroid. That sounds far-fetched, but we've been analyzing asteroids, and they have some interesting compounds on them, including amino acids and small sugars. So it is possible that life had an extraterrestrial origin. What's gaining momentum, though, in evidence accumulation is undersea thermal vent. Undersea thermal vents provide both the space and the opportunity for aggregates to form. In this case, you have energy, which is volcanic energy, bubbling out into the ocean. That interaction of volcanism and the ocean water forms an entire cocktail of molecular compounds, and some of them are biochemicals. They're random biochemicals, but they're biochemicals. And they could have organized themselves randomly into aggregates that then moved forward with chemical selection to the first life forms. So these three possibilities are constantly being debated, but right now undersea thermal vents are getting the most attention. So let's take a look now at what research has been done. By the way, we're looking on Mars to see if Mars has any evidence for life, that maybe we're missing it. Maybe Martian life is unicellular and below the soil surface. We think that the Martian current conditions are probably a lot like it was on the early Earth. This is the Cape Verde crater on Mars. There could be water in the bottom of that basin below the surface of the soil. Panspermia is the concept that, biochemicals or cells or something from an extraterrestrial originated life, but gaining momentum is this concept that deep sea thermal vents did that job. Now, the tidal pool scenario rests on the concept of a reducing atmosphere. Reducing gases are gases that are things that are not oxidizing gases, so they don't have oxygen gas mixed with them. This would be carbon dioxide, methane, hydrogen gas, water vapor. Those are all potential reducing gases. Ultraviolet light hitting those and lightning hitting those converts them to small monomers. These are small carbohydrates, amino acids. It's like air pollution would accumulate in the air. This has been tested in the lab and it works. The rain would have dropped those into tidal pools. So from the sky, rain would have been bringing down food molecules like manna from heaven to the ground. Those would have collected in small puddles, and periodic drying and re-wetting of the puddles would have caused polymerization of the monomers into polymers. This has been tested and it's been shown. Sidney Fox did a lot of extensive testing on this. Proteins, they're actually called proteinoids, do randomly form when you mix amino acids into pools and then rapidly dry them. The difference between a proteinoid and a protein, though, is that proteinoids polymerize randomly, whereas proteins are always bound by peptide bonds specifically between carboxyl and amino groups. So far, no one's shown exactly how proteins could have formed abiotically, but it's been shown that clay will enhance polymerization and cause organic molecules to polymerize in specific ways. So it is possible that we had a reducing atmosphere origin and then tidal pool accumulation of stuff, but ultimately the tidal pools would have been drying and re-wetting, and that would have caused the polymers accumulating in them to aggregate. What an aggregate is a clump of polymers that are all balled up together. They're called, aggregates. Now, a specific kind of aggregate is the microsphere. Microspheres are technically just made of proteinoids. Coacervates are technically aggregates of fats and carbohydrates. Well, it could have been a combination of microspheres and coacervates accumulating biochemicals, which would have been developed in these temporary, tidal pool or puddle areas on the planet. And basically, these microspheres could have developed what are called emergent properties. In other words, long-lasting microspheres might have microspheres do randomly butt off and make little microspheres, just like soap bubbles do. It may have been that may have been the first strategy of reproduction of the original life forms until actual life showed up on Earth. The reducing gas tidal pool scenario is summarized in this table. You have atmospheric gases in the form of reducing gases being hit by ultimately ultraviolet light, UV or lightning, and then that forms tidal pools full of cocktails of chemicals. The cocktail of chemicals would have led to the first polymers by drying of the tidal pools. That would have led to cell-like structures called aggregates, and that would have evolved into the first cells. This is the part, though, this is the part no one's been able to duplicate. All of the other steps have been duplicated. No one knows how this step happened, though. So between 4,600 and 4,100 million years ago, something happened and led to the first cells. We have evidence of the first cells then. The scientists that have done the most research on this are Alexander Oparin, the Russian scientist who probably did the first research in publishing 1924. J.B.S. Haldane, a British scientist, developed the same idea around 1929. Okay, Oparin was a Russian scientist. Too bad they didn't get together and talk. 1953, Miller and Urey did a series of experiments and did verify that a reducing atmosphere would actually form monomers. Other scientists have shown that those monomers will randomly polymerize to form polymers, which can lead to coacervates or proteinoids. This is Stanley Miller here, and this is the basic strategy of his experiment here. He used basically sterilized chemicals in a spark chamber, hit them with electrical spark for about a week, and then tested the chamber, and he found carbohydrates, small amino acids, all kinds of things. So ultimately, the overall pattern of evolution on Earth is that the Earth cooled between 4.6 and 4.1 billion years ago. And the life originating processes were probably already beginning to start at the end of that. Somewhere between 3.7 and 4.1 billion years ago, life actually gets underway. Don't know exactly how. It might not have been by volcanic vents, and it might not have been from tidal pools. It may have been something else. But at 3.7 billion years ago, we have cell-like structures that led then to the first cells. We think that those first cells would have probably used RNA as their original biological storehouse of information, but that over time, DNA evolved because it is a much more stable, long-lived molecule than RNA. So by 3.5 billion years ago, we're thinking DNA was probably the code book for storing biological information. Ultimately, the evolution then would have been for how do you power yourself. The original evolutionary power source would have been glycolysis. That is a simple breakdown of sugar. It's anaerobic. It didn't require oxygen. The atmosphere back then didn't have oxygen 3.5 billion years ago. And ultimately, these original cells would have been then powering themselves with breakdown of simple carbohydrate compounds. They would have been storing information in terms of RNA, but moving towards DNA. And they would have been manufacturing small organic biochemicals, which are proteins and other carbohydrates. Enzymes would have been evolving. The storage of enzyme production information in RNA and DNA would be going about. Ultimately, though, this is all leading towards membrane-bound organelles. We think by about 2 billion years ago, membrane-bound organelles showed up, and that's the first eukaryotes. You can't build multicellular bodies without having internal cellular complexity, and eukaryotes are a lot more complicated than prokaryotes because of those internal membrane-bound organelles. So on Earth, what we have now are three grand three grand types of life forms. We have prokaryotic bacteria, which are everything from E. coli to pneumonia bacteria. Most of the stuff that's growing in soil is prokaryotic bacteria. They lack membrane-bound organelles. They have a phospholipid membrane with what are called ester lipids, and they have very simple gene control systems. They use glycolysis, all of them, for their energy pathway, but some of them are also aerobic, and they have other enzymes too. Other prokaryotes evolved, though, that we call archaeans. They also lack membrane-bound organelles, but they have more complex gene control systems. That allowed them to do stuff genetically that the prokaryotic bacteria could not do. They also have ether-based lipid membrane systems, and they use glycolysis as their energy pathway also. Ether-based membrane systems, they don't use the ester bond to bond the fatty acids to the glycerol. They use an ether bond, which an ether bond is a carbon with a double bond. An ester bond is a carbon double bonded to an oxygen, single bonded to an oxygen, forming a bridge between two compounds. So at this point now, here and here, we can have another organic compound here and another one here, and this goofy cluster of carbon, double bond oxygen, single bond oxygen can bond them together. An ether bond is simply an oxygen single bonded to a molecule and single bonded to another molecule. So this is an organic molecule and this is an organic molecule. Water is the most basic ether bonded chemical. Water is just basically oxygen single bonded to two hydrogens. When you have, in place of those hydrogens, if you have other organic molecules, you have an ether bond between those organic molecules. Why archaeans use ether phospholipid membranes, we don't know, but there may be, that may be figured out in the years to come. Eukaryotes have complex genetic control systems. They have ester phospholipid membrane construction. We have linear chromosomes, and we use glycolysis and other aerobic energy pathway systems. We also have membrane-bound organelles. So that concludes the chapter on patterns of evolution. It's a rather short one. It just talks briefly about the different overall patterns of evolution, or the story of evolution on Earth.